Testing DNA Evidence by Daniel Blevins. In this PowerPoint, we're talking about the DNA structure, DNA and law enforcement, and also the DNA testing backlog. The DNA structure. DNA is a double helix. It is made up of molecules called the nucleotides. The nucleotides contain phosphate groups, sugar groups, and a nitrogen base. The nitrogen base, there's a total of 4 nitrogen bases: adenine, guanine, cytosine, and also the thymine. When thinking of the DNA structure, think of it as a ladder. The two sides are the phosphate group and the sugar group, and they can create the sugar-phosphate backbone. And then the nitrogen bases are the steps in the middle. Adenine and thymines go together. The guanine and the cytosines go together. Since DNA is unique in everyone unless they are identical twins. What creates the uniqueness is the order in which the nitrogen bases go. DNA and law enforcement. DNA is used in law enforcement every day. DNA identifies criminals that have committed a crime. When the DNA goes to the lab to become analyzed, they find matches from either the DNA database or also look at the DNA profile of DNA evidence that they have collected from potential suspects for that case. DNA has helped convict criminals, clear suspects, and also exonerate people who have been wrongly convicted. Collecting DNA. When collecting DNA, it is important to stick to the protocol, to follow protocol. Make sure to wear gloves and use disposable instruments like cotton swabs. It is also important to place evidence in paper bags or envelopes. Never place DNA evidence into a plastic bag because plastic bags can contain moisture, which will then contaminate the DNA. And also find the chain of custody. Follow chain of custody. Chain of custody is a chronicled document that records the sequence of custody, control, transfer, analyze, and disposition of evidence. The chain of custody identifies who has had the evidence and when. So if for some reason something happens to the evidence, they can go back to the last person that has it to figure out what has happened. This is an example of the label you would put on the envelope or also the paper bag. When the DNA is collected, it is then sent to the lab to be processed. There are 5 steps to processing DNA. Extraction, which is the process of releasing the DNA from cells. Quantitation is determining how much DNA there is. Amplification, process of making multiple copies of DNA. Separation is the process of separating the amplified DNA. And then analyzing the DNA by comparing the DNA to a DNA profile from either the database or DNA evidence collected from potential suspects. The polymerase chain reaction is used during the amplification step of processing DNA. It creates billions of copies from a small amount of DNA and only takes a few hours. There are 3 steps. The first step is separating the 2 strands of DNA by heating the starting material to 95 degrees Celsius. The second step is reducing the temperature to 55 degrees, which allows the primer to anneal to the template. Step 3 is processing the temperature to 72 degrees. The DNA polymerase begins adding nucleotides onto the ends. It only takes about 5 minutes to do 1 cycle, and you only need to do 25 to 30 cycles to reach enough DNA to be analyzed. Right here, it just is a diagram to better show the steps of the PCR. The DNA backlog. DNA backlog. There is no set definition for what the backlog is. The first definition is DNA evidence that has not been analyzed in 90 days. Second definition is DNA evidence that has not yet been analyzed. The DNA backlog has increased 95% in the past 6 years. State and local governments have received $1 billion in the last 15 years to try to reduce the backlog. The reason that there's a backlog is because since DNA has become such a commonly used tactic in solving crimes, there has been an increase in requests. They get requests from current cases that are trying to be solved, cold cases from when DNA wasn't really around before the 1980s and even after, because over the years DNA testing has had a lot of technology advancements, and also post-convicted cases to try to help people that are wrongly imprisoned. How can you solve the problem of the DNA backlog? Then NIJ's Forensics DNA Backlog Reduction Program. The purpose of this program is to try to reduce the average time to complete analyzing of the sample. They want to increase the number of DNA samples being analyzed, and the main goal is just to reduce the backlog of DNA casework. The program provides grants to state and local governments that need help. There are also two types of technology that scientists have been creating. The first one is the magic box. The purpose of the magic box is to have a DNA processor to do the job in 90 minutes. They put it into the actual area in which investigators are working. And the investigators can actually process and analyze the DNA by themselves instead of sending it to a lab, which can take up to 14 days to actually begin processing. And it takes about 24 to 72 hours for it to actually be processed after it has been sent to the lab. And then there's also a portable DNA testing. The purpose of the portable DNA testing is that it can test the DNA and be analyzed right then and there for cops. So you would take the swab from the mouth of a suspect, mix it with a chemical, and warm it up. It only takes about 4 hours to do instead of the 24 to 72 hours. So throughout using technology, we can try and reduce the backlog as best as we can. So with money and technology, with both of those, we can eliminate the backlog in DNA testing.